Again, thank you everyone for coming. My name is Arissa. I'm the Outreach Associate with the Coconut Rhinoceros Beetle Response Program. Myself and then Keith Weiser, who's also here with the Coconut Rhinoceros Beetle Response Program. We're going to be manning the tech and doing the background stuff here. And then we also have Sana, who is with the CRB Response, and she's going to be giving some general information about CRB before we go ahead and open up the floor to our speakers today while they share about their research for CRB. So we'll go ahead and hand it off to Sana to share on the coconut rhinoceros beetle background.
Thank you, Arissa. My name is Sana, and I'm the Other Outreach Associate for CRB Response. So just some quick background. The CRB was first found on Oahu in December of 2013, and then it was only found on Oahu for almost 10 years before it was found on Kauai, Maui, and Big Island last year in 2023. And just some quick identification. The coconut rhinoceros beetle is an all-black 2-inch beetle with a horn as an adult. And then it's when they're adults that they feed on palm trees and other crops. And then the larva is this white larva that can get up to 4 inches. And that's where you're going to find in breeding material, which can be any decomposing plant material. And I'll go in a little bit more into detail on what that is in a little bit. So the adults, they will feed on their primary food sources are going to be coconut, royal, date, and fan palms. But we have seen them go, if those food sources are not available, to go for eat things like banana, sugarcane, hala, and even kalo, sago palms, pineapple, and ferns. And so when the adults feed on the beetles, they will fly up to the crown area of the palm, and they will bore in to feed off the juices and the sap in the heart of the palm. And that will leave these 2-inch boreholes. And when the palm fronds grow out, they're going to have this V-cuts. And this is the damage you will see in coconut, but there will be some sort of other. You can look a little different on other palms, but you're always going to see these 2-inch boreholes. So on Oahu, CRB is pretty widespread, and most areas have some sort of population of CRB, but we have some areas that are have more beetles than other and also see more damage. So that will be areas of Central, West, and North Shore. And that's when we start to see this kind of damage. So ultimately, multiple feeding events can ultimately lead to palm death. And so this is from a park in West Loch on Oahu a few years, or in September 2022. But this is kind of common sites now in those areas that I mentioned earlier. So as I said, adults are the ones who are feeding off the palms, but they will not stay in the palm their whole life cycle. So after they feed on the palm, they going to go and try to find a breeding site. And then that's where they're going to lay their eggs. And then they will go through 3 different larval instars before they pupate and turn into the adults. And in these earlier life stages, they actually spend more time in these life stages than as adults. So that's where you find them in decaying plant material. So that can be things like chipped green waste, or also known as mulch, or whole green waste. It can also be found in logs, standing dead trees, compost, and potting mix, as well as potted plants. So what can you do? First thing is to inspect all potential breeding material or host plants and not transport CRB. So if possible, not transporting any of that possible breeding material, but if you have to transport, make sure that it's clean before you move it. And then manage high-risk material. So manage that breeding material. We have different best management practices on our website. And then stay informed and spread the word. So our website, where you can find a lot more information, is crbhawaii.org. You can also email us at info@crbhawaii.org, and then you can find us on social media at CRB Hawaii. And with that, I'm going to turn it back to Arissa to
All right, thank you, Sana. So now we're going to go ahead and pass it on to Dr. Cheng. So I'm going to go ahead and introduce him while he pulls up his slides. So Dr. Cheng is a professor and extension specialist in CTAR at UH Manoa with 21 years of research experience on biology, ecology, and management of turf grass and landscape pests. So today he's going to be talking about chemical and biological controls for CRB.
Hey, thank you, Arissa. Let me go ahead and share my screen here. Okay, share. I'm gonna get rid of the floating meeting controls. Okay, so. All right. So today, well, we only have like 10 minutes for each person, so I will just have to power through all my slides. But we will be talking about research update on chemical and biological control of CRB in Hawaii. And I want to acknowledge my former and current lab members. Not everybody contributed to the research I will be presenting today, but we work as a team. So I do want to acknowledge that upfront. And I had a few actually introduction slides on CRB, but then now I see why Arissa asked me to remove those, because the introduction was much better and more comprehensive than my introduction slides. But let's just go straight to the research I have been doing, my lab has been doing. So we have focused on systemic insecticides for the chemical control, direction, and also we work on entomopathogenic fungi and nematodes on the biocontrol direction of CRB control. Me and Mike, we started working on CRB together since 2014, right after the CRB was confirmed on Oahu. And for chemical control, initially, we had difficulties working with the CRB, lab assays, because we had to rely on wild caught, the trap caught CRB specimen to actually run experiments. We can only get, 30, 40 here and there. That's why we were only able to run small batch, experiments in the beginning. That was before the CRB colony was established on the 5th floor of Gilmore, a few years later after CRB was confirmed. But long story short, we tested a lot of insecticides, mostly systemic insecticides, to work with, in lab assays. And as you can see, we set up messengers and beetles, and we feed them with different chemicals, insecticides, and treatments. And we observe their, condition, whether they are killed, whether they are paralyzed due to the treatments. And in summary, quick summary would be, we tested about a range of insecticides, and in the end, based on the lab assays, we identified that imidacloprid and acifed, these two insecticides, were giving us pretty quick actions in terms of killing the CRB adults. So we need that quick action, because as the former speaker mentioned, and the CRB adults only get on top of the palm trees and stay there for 3-4 days while feeding. And we do want to utilize that window when they actually ingest the chemicals, on the part where they feed on. Then if they are negatively affected within those 3-4 days, that's the window that we want to target using this approach. Instead, utilizing the systemic insecticides, we do want to have that quick action. So this is a quick, summary, a lot of results, but I just want to give you guys a overall impression that for imidacloprid, we were able to see mortality after 2 or 3 days. And even within the first day, within the first 4 hours, we begin to see some negative impact on CRB adults using imidacloprid and acifed. So this gives you a quick general impression that, okay, so now based on all these lab assay results, and we are comfortable moving forward with setting up a field trial. So that's why we started our first field trial, relatively long term, actually for about 3 years, at Ilocos Point side, on the Ever Beach side of the island. You can actually see this actually was the case. He has been working with us in the kind of relatively early stage as well, and we were using the trap injection approach to deliver the systemic insecticides, setting up the field treatments over there. We used 125 coconut palm trees in that area, and this was also in collaboration with Arborjet as well, and also funded by the USDA grant later on, and HISC proposed grant later on. So this is the standard data collection that we have been doing over the years, and we essentially borrowed this idea from Palau, collaborator, and why we it's not like we simply want to copy what they do, but we also want to be able to potentially compare our treatment effects versus other regions where they have treated CRB, and we have been dealing with CRB, infestation, and potentially compare our results with other people's research results. So we essentially looking at 2 variables: the overall damage and also the top 4 fronts, where any new damage will show up initially on any palm trees. So those are the 2 variables we measured, all by visual. But then we had 2 people doing the visual at the same time, so to potentially reduce the potential bias from same person, right? So after 3 years, what we got out of this, overall treatments in that given area is, although acifed and imidacloprid worked almost equally effective in lab assays, and then in the end, imidacloprid was kind of like the winner based on the field research that we have done over there for about 3 years. And this picture, I always have this picture, and I kind of like always tell kind of small joke that we did not grab a dead beetle and put it on the grass and take the picture. It was actually the dead beetle found on the grass where we treated the palm trees. So it's not a staged picture. So it's a direct indication that beetles were killed by our treatments. Another indirect indication about the area-wide treatment is when we compare to an area where there was no treatment. And then as you can see, this is the blue line about several miles away, and we got the data from the CRB response team. So the CRB population density was slightly increasing, but with our treated area, the CRB population density reflected by the trap count number was decreasing to some extent. And then that's another indirect indication CRB treatment with this insecticide, systemic insecticide were working to some extent, at least if you are able to treat the CRB on a relatively large landscape scale. So after that, we conducted additional lab assays, and when more and more systemic or low risk insecticide became available for us to test, and also at this time we had the colony established already, and we were able to run better designed larger experiments in the lab assays, and we further identified that 2 additional systemic insecticides, which are dinotefrin and abamectin, both were working almost effectively, equally effective in lab assays. And then we decided, okay, so we should take another look at additional, field trial. So we did that. So this was a brand new field trial. We started in December 2019 and concluded in mid-year 2022. So we had 4 treatments, as you can see, both imidacloprid and acifed. We just want to retest them and make sure they still work. They work, equally, compared reflecting our previous research field trial. And also we included these 2 additional pesticides which were effective in our lab assays. And then we collected the palm, fronts, and also documented the CRB, damage level and everything. And we again found dead beetles on the ground where we treated the trees, not staged pictures. And also, interestingly, we were doing this front feeding experiments. We basically collected the fronts from those palm trees where we treated with trunk injection and bring them back to the lab, chop them into small pieces, and feed those small pieces with the CRB adults reared by the colony. And we want to see what's going on, whether they can be actually negatively affected, to essentially to confirm that the CRB was actually killed by the treatment that we installed. So that was the case. And then based on the front feeding experiments, which I think Keith just mentioned before the webinar, that he will be doing a similar research as well. And we were able to see that even 7 months post-treatment, so we were still seeing that negative impact of the front feeding to the CRB adults. So all this research was kind of like the leading point for the CRB response program to start treat the CRB the coconut palms with the trunk injection of imidacloprid. I think you guys probably know more details. I'm probably treated over maybe around 4,000 coconut palms so far. But then that's how we work on this field trial. And this is a very new brand field trial. We just started last week back in May just earlier this month, last week. And we included 39 palm trees at Ted McLennan Golf Course again. Those 39 palm trees, they only have moderate damage at this point. So then we were testing, we begin to test additional new systemic insecticide and one biopesticide. So this is just brand new, and we just started the treatment and begin the data collection. This will run for at least 1 year, and depending on the results, and we will decide whether we will be continuing a second year field trial with this new research. Okay, so that's the chemical control part. For biological control part, we mainly look at the two groups of biocontrol agents, entomopathogenic nematodes and entomopathogenic fungi. So for nematodes, our approach for actually both of these two groups of biocontrol agents, our approach is to collect locally occurring strains. The reason being that we have very strict regulation on importing biocontrol agents from the mainland or from other countries into Hawaii. Even they can be commercial products already, but then we don't necessarily have that easy access to those commercial products. Then we begin to work on the biocontrol agents by locally collecting them and test them one by one, and select the most virulent ones and further narrow down to refine the test and see whether they can be effectively used to control CRB. If they are effective, then, and also because they are already here, then we are not bringing anything new, then we can potentially make use of them. That's kind of like the overall concept of this biocontrol research we have been doing so far. So for nematodes, we did a range of tests, and unfortunately, we did not find any very effective ones, say over, I call it over 70% or over 60% mortality effective. If we have that in lab assays, we did not have that kind of high mortality in lab assays. So then the highest we were able to get was about 35%. This is also not consistent. So then we decided to kind of spend invest more time and effort on the animal pathogenic fungi side, where we got a much better result in terms of the mortality. So initially we collected about 60 strains from all over the island of Oahu, and we extract them. And we had mostly Bavaria and Matarizan species. And then we narrowed down to about 15 strains where we had over 60% mortality. As you can see, we were running the preliminary screening at the PQ, Sand Island facility. And then we did further refine the test on those 15 strains and narrowed down to about 4 strains where we had over 80% CRB mortality over a period of time. This is to give you a quick idea. We coated them so that we know what we are talking about. We coated them and maintained the colony in the lab. So then we also were able to run small scale field trial, in different locations, in 2 or 3 locations, I believe, and we were able to get about 40%, in some cases about 60% or 70% mortality using our locally collected strains and also the commercial Bavaria bassiana, the botanic garden strain as well. So these are the kind of like the direction where we will be hopefully looking at in the biocontrol direction in terms of between entomopathogenic nematodes and the fungi. We definitely had a better result coming out from the fungal pathogen side. So a quick summary. I don't know if I took more than 10 minutes, but basically on the chemical control side, we identified 2 chemicals that were 4 chemicals effective in lab assays, but then in the field trials, really it was only imidacloprid and a little bit with the acid that were giving us treatment effects on a large scale situation. And then for the fungi and for biocontrol agents, we identified several locally collected strains, Matarizan strains, and also the botanic garden was pretty effective as well. And the next step would be potentially to mass produce these locally collected fungi, and hopefully we are able to go through all the regulations, hopefully, and distribute to local, people who can use them. And also we will be continuing the current, lab and field research with additional insecticides and potential additional biocontrol agent selections. That's all I have. I always have the standard disclaimer because I use the chemicals. So I just want to make sure everybody is informed that, we do not endorse any of those, but we provide information based on the research that we have conducted. Okay, any acknowledgement for funding agencies and also collaborators who contributed to this research in terms of CRB biocontrol and chemical control. That's all I have. I'm going to stop sharing. Let's see. How do I get back to that?
So I'm going to be next, right?
Yeah, I think.
Okay,
but
I'll
I
start.
need to. Can you push me out by sharing your screen? Because I'm not getting back to the bigger screen. Oh, okay. All right.
There you go. Okay, so
Okay,
now
you.
we have Mike's screen. Thank you. So next up is going to be Dr. Mike Melzer. So
yeah, you got
perfect. So I'll go ahead and introduce you while you get that all set up. So Dr. Melzer is an associate researcher at the University of Hawai'i at Manoa who specializes in agricultural biosecurity. His program's focus include preventative pest surveys, response to pest and pathogen incursions, pathogen diagnostics, and development of clean plant germplasm. So Dr. Melzer is going to be talking today about genetic tools for CRB management.
Yeah, it might be if I get this. I never get this right. And I apologize for this. I'm going to, does anyone want to give me a suggestion here? Which one it is? I always get this wrong.
Maybe on display settings.
Display settings. It's usually up here. I thought if by turning off my one computer, I would be able to, or my one screen. Let's see. I'm going to stop this share. Let's go with this one. Nope, same thing, right?
Perfect. Nope, that's good.
Oh, it's working. Okay. Okay. Well, I can only see the preview. Well, anyways, good afternoon, everyone. My name is Mike Melzer. Sorry for the technical issues. I'm good for one of those, at least one of those in every presentation I do. I'm going to expand a little bit on what Dr. Cheng was talking about and complement some of his work and look at some of the other work we've recently been doing in our lab. And mostly what we're trying to address here is, especially with this few slides, is our short and long-term management options for CRB and what our lab is looking at right now. So I think Sana did a good job introducing our two, the two main life stages, the adult and the larva. Larvae are detritivores, so they're in the soil and green waste, and that's where they're breeding. Whereas the adults are feeding up in the palm trees themselves, feeding up in the canopy as sap feeders. So there's two different approaches to tackle each of those life stages. In terms of short-term management, basically what we've been, am I on the right slide or am I on the preview? Yeah, the short term, basically the one option we can look at for the larva is breeding site treatments. And in terms of chemical control, Dr. Cheng mentioned a few that can be used. He was looking more at trunk injections, but they also work somewhat well on larvae as well. So imidacloprid has shown demonstration, whether it's something you can till into your mulch or include it as a chemigation. We have found that pyrethrum and pyrethrins do work for CRB, so these are more contact insecticides. However, they're probably some of the pyrethrum and pyrethrins in their organic nature, those are the ones that basically are coming straight from the chrysanthemum plant. They don't tend to have the longevity in the field. Whereas pyrethroids, the synthetic versions, usually of one of the pyrethrins, they tend to have better longevity. They've been designed and manufactured to have better longevity. We've also found that sulfurofluoride works as well for treating breeding sites. And again, this is an endorsement, but this is a large-scale treatment fumigation that was done over on Kauai. And we have tested the parameters as to what levels of sulfurofluoride. And for those of you not familiar with that chemical, that's the same one they use to tent your houses with for termites and other pests. And so we have determined that, and I think there is a special local needs permit underway based on some of those results that will allow sulfurofluoride to be used as treatment for CRB breeding sites. Additionally, we've identified some physical parameters for killing CRB in their breeding sites. One is thermal, if you can get the temperature over 130 Fahrenheit, or if you can get them, if you can freeze it, that's a bit more challenging in Hawaii. That's going to devitalize them. Chipping and grinding is typically going to devitalize CRB. Submersion, particularly if you can do it in brackish or salt water and say a tidal inundation zone, that's going to be helpful as well. Or just burying the material deep enough so that hopefully the CRB cannot detect it and lay eggs. And then we're always recommending spreading thin. And how thin you do it usually depends on how much moisture content is in the area. The wetter it is, the thinner you have to spread it. So windward sides of islands, you can get away with maybe an inch or so. On drier sides, you might be able to get away with a couple of inches as well. And the spreading thin has the additional benefit of allowing other predators and so forth getting access to any grubs that might be present. When it comes to other short-term management options, then we're looking at the adults, and there is the host palm treatments. And as Dr. Cheng mentioned, injection in root drenches, and we've adopted his approach for imidacloprid and acephate in the field. And that is can be done either directly through an injection or for small trees, or if you don't have the equipment, a root drench where the roots uptake the chemical and translocate it up into the canopy. Sort of a bit of an expansion. So as Dr. Cheng alluded to, like in Kapalina, where he did his initial trial, there's where we did an initial, landscape level treatment. So almost 1,000 trees were treated there. And we can see on the graph below, there is that sort of reduction in trap catches in that landscape level treatment. And so we think that's, probably a result, and probably also, as Dr. Cheng showed, after about 7 months or so, well, that's how good it was good for. And we started to notice some upticks after that. We typically recommend the trunk injections when you're doing good breeding site management. You need to basically stop. The injections are going to work best when you're controlling the population in other methods and not just relying entirely on the injection. The other approach that we're looking at, and so we're sort of complementing Dr. Cheng's lab, we're looking at some more of the canopy approach, canopy treatments. You're going to hear a bit about that more from Dr. Jenkins, but again, looking at the pyrethrums and pyrethroids, and then obviously sulfurofluoride is not going to be used in the canopy. That's a mistake on my part. Apologies. I don't think we're going to be treating the canopies with fumigant. How we're looking at for the ground-based application is working with, in the future, what we'd like to do is work with arborists, tree trimmers, and other pest control companies to probably subsidize some of these treatments. As we finalize and identify which are the best treatments and what additional regulations have to be overcome, whether it's some sort of labeling requirements, but we're looking to identify which chemicals are going to be the best for some of these, some of the pyrethroid and pyrethrum-based chemicals that are going to be best for applying in the canopy. Typically, it's going to be pretty easy for a short palm tree, or if you've got a bucket truck where the trimmers who are up in the canopy and potentially who are climbing up, they're going to have a bit more challenge with treatments. I'm not going to talk about it today, but we are looking at some options for them as well that would allow, but there's going to be additional regulations for that. So we're looking at, we're looking at experimenting with an approach that people who are climbing the trees can do applications to the canopy as they're doing their regular maintenance. And of course, this is going to be the increase in difficulty of adoption. So one of the things we're looking at right now is the pesticide persistence in the canopy. And so we're evaluating 5 different chemicals plus control just to see which ones last the longest up there, are going to have the best bang for your buck at this point. So these are the 5 that are being evaluated at present. We basically are 1 month in, and based on that 1 month, number 1, number 2, and number 3 tend to work the best so far 1 month in. Suspend is maybe one nick behind, and then the organic option, which is the evergreen, the pyrethrum-based product, works very good initially, but it sort of, the efficacy tails off fairly quickly. So once we've got better data on this, and that's going to allow us to make better recommendations for industry in terms of what product is best and what interval, and it will also help us identify what registrant for these products we can work with to adjust any labels as necessary. Dr. Cheng also mentioned about biocontrol and identifying and registering any localated. This is we're starting to get into long-term management. These are the tools that are not quite ready to go, but we hope to have for on a longer-term scale. There's some regulatory hurdles that are required for any biocontrol work. And then, we have the known ones, as Dr. Cheng mentions, the Metarhizium and Bovaria. Trying to identify the best one so that what does come to a registration, we're using the best strain out there for efficacy. There's also potentially going to be some work for foreign exploration, either with ourselves or with partners in the natural range. There's also been discussion about working to import a nudibvirus that's been used for biocontrol elsewhere in the world. At this point, we don't necessarily have the biocontrol facilities to do this work, but hopefully we'll be addressing that in the future. And the other approach that I'm going to spend a couple of seconds talking about is development of an RNA interference vector. I'm not going to get into the science of this at this point, but some of you may or may not be familiar with this approach as it's called RNA interference, but you're probably familiar with it as things like gene therapy or other medical uses. There's basically RNA interference is being used for cancer treatments, any congenital diseases or any other genetic-based diseases. So it's getting fairly common in medical applications. In agriculture, it's starting to emerge as well. Bayer recently generated a technology for targeting a corn rootworm using RNAi as RNA interference as an approach. So we've been looking at this. Some of us are also familiar with the transgenic papaya. It wasn't known at the time, but the mechanism that is responsible for resistant papaya is actually RNA interference. It's just in the animal world, it's RNA interference. In the plant world, it was known as post-transcriptional gene silencing, but it's generally the same principle. We looked at RNAi for CRB several years ago, maybe in the 2019, 2020, and we found that it works. We can basically target some essential genes for CRB. These graphs aren't necessarily helpful for you guys, but we identified 6 targets that basically when we target those in different life stages, whether it's adults or the different instars, the mortality increases. So as those lines go up, that shows mortality after 1 week and 2 weeks. And in general, we were reaching 100% mortality after 2 weeks. We also looked at to see how small of a dose was required, and it's quite small. It's probably less than 10 nanograms. So we're probably, well, anyways, we're looking at pretty small doses required to devitalize CRB using RNA interference. The biggest issue we had is delivery, and why we put this thing on the back burner is because we tried all the known ways of delivery as a sort of maybe what you could imagine as a conventional pesticide, and none of them were particularly effective. But one thing we're starting to look at again is delivery through a virus, very similar to how gene therapy is often delivered, which is usually through a viral vector. A study not too long ago was published on using RNA delivery to combat an important insect in agriculture responsible for a citrus disease or spreading a citrus disease. But importantly, just in the last 2 years, a group in Australia and Taiwan identified a vector in Coconut Rhinoceros Beetle that could be useful as a viral delivery mechanism for the RNA interference. And so this approach would be basically akin to the medical use and veterinarial use of adenoviruses, some of the adeno-associated viruses, and other viral vectors that are used in gene therapy. So this is something, far away, There's a lot of regulatory hurdles with this, but at this point, it would just sort of be a proof of concept. There's a lot of advantages to using this due to the high specificity and no non-target, the theoretically no non-target effects like you might see with conventional synthetic or other pesticides. I think that's what I've got. I want to thank my lab or our lab. This is a really old picture. There's been a lot of turnover, but I need to get some newer pictures. But in any case, I think that's all I've got. Thank you.
Thank you. So now I'm going to go ahead and turn it over to Dr. Jenkins and Mohsen. So if you guys can share your screen, and I can go ahead and start with intros. Okay.
Yeah, Mohsen, you go ahead. Okay, sorry.
Oh, thank you. Okay, yeah. So up next we have Dr. Daniel Jenkins. So Dr. Jenkins is an agricultural engineer with 20 plus years experience developing new technologies for detecting and controlling pests and disease organisms, and for supporting management of agricultural and environmental resources. And then Mohsen is an electronics engineer and a PhD candidate. His research at CTAR focuses on the early detection of CRB using camera traps and aerial pesticide applications for CRB pest control. So Dan and Mohsen are going to be talking today about CRB delineation and control with embedded surveillance, artificial lighting, and aerial pesticide application.
Okay, hi everyone. I'm Mohsen Paryavi. As Arisa said, I'm an electronic engineer and PhD research student in Dr. Jenkins' lab. And today I'm going to talk about our trap camera board. Dr. Jenkins and my research advisor and I designed an automatic Coconut Rhinoceros Beetle trap camera board. And here you see, if I can click here, see the top part of our camera board, which has a digital camera and microphone. And also it has you see the GPS antenna and the cellular modem there. And also it has some energy management control and programming part. So the way it works, the camera takes the image of the trap, and also you can use the microphone, and all the data will be, and also all the data of the camera, microphone, or also the GPS data can be fetched from the GPS satellites, and all the data can be sent through the modem that you see to our server. And the energy management part is for having an automatic and sustainable energy device where we can have a solar panel, and the energy management part will use that solar panel to charge our compact LiPo battery. Also, you can program the board with the tiny computer there, which is called microcontroller, that you can program it to how many images per hour you want to send to the server. So this is the top view of the camera board, and the bottom view is you see the white LED and also the camera. So the white LED illuminates the trap cup of the panel traps before it takes the image, because mostly CRB is active through the night. So in this slide, we see how we can wrap the board that you saw in a custom enclosure that Dr. Jenkins designed, which is weatherproof because all the traps are outdoors, the panel traps, and we wanted to have a weatherproof enclosure. Also, you see the antenna. So it's like your cell phone, it takes the image and send it to the server. And here you see how the enclosure which wraps the camera board is mounted on the panel traps cup. So the bottom part is the cup of the panel trap. And then if you look carefully, you see the solar panel there at the top part. And there is a cable that connects the solar panel to the camera board. So during the day, the battery gets charged, and then we can have it sustainably deployed for as long as the camera board works. So typically, you know, we had it for most of the boards worked for a few months to a year. The one, and then on the right side you see the camera board, how we mount it to the trap cup, and then how it faces the trap cup, and takes the image of the trap cup. And it's very complex design. And how the program works is the board is most of the time is in idle mode, because we don't want to waste the energy. And then it wakes up, let's say after, as you program it, but in our experiments, every hour it wakes up from deep sleep mode. So, and then takes the image, gets the GPS data from the satellites, and to provide the GPS fixes or the location of the trap. Also, it has each board is programmed with trap name, and then also it takes the image, and if it's night, you know, it will turn on the flash LED and sends all this data through a cellular network like AT&T to the server. And then in the server, we have it's a web server with like a website, and which has the image gallery, and you can see the traps data, which is the trap name, also the time, date, and you see which traps are having how many beetles. And this is very useful for early detection of beetles. For example, in Big Island, our camera boards could we sent we deployed a few camera boards in Big Island, and then the first adult beetle was found with our board. Also, we had some early detection on Hawai'i, and also in order to field test but before that, in order to field test our custom designed trap camera board, we deployed 10 camera board to field, which was reporting every hour, and they were working. They were sending data in the first eight months of 2023, from January to August, and so this is very useful because the yellow dots that you see in the picture are the deployed trap camera boards, and the black dots are all other traps around Oahu that CRB Response team monitors. And our camera boards are useful in, other than early detection, it's very cost effective, and people don't have to go transport to the trap location, which is classic monitoring, to detect a beetle. So this is the camera boards were efficiently was sending the data of the trap to our server. And in doing this research project, in the first 8 months, because we had images every hour, with data analysis, we illustrated that the beetles are crepuscular, which means they are active mostly during twilight, and mostly 2/3 of catches were happened 3 hours after sunset, the CRB catches. So I think that was my slide. Thank you for your attention. Dr. Jenkins will continue.
Good. Yeah, thanks. I'll jump right into it. Let's see. Can you release the screen, Mohsen?
Yes.
Thank you. Okay. Okay. So can everybody see my screen? Okay. I'll just jump into it. So, so kind of following up, I kind of want to share some results about evaluating artificial lighting to kind of improve the trap catch, so kind of in tandem with the work that Mohsen already described. So we kind of actually maybe half a dozen years ago or more kind of developed a kind of programmable lights with different color LEDs, and you could program them, turn on different times of night, you know, and modulate in different ways to kind of evaluate actually for a different kind of insect with biosecurity implications. But so kind of, and we tried to use these in the lab when, you know, before CRB became a really serious problem, and kind of the results were pretty inconclusive. So when you dig adults beetles out of the, you know, the kind of nesting boxes, they just basically want to avoid all light and go back and nest, right? So so anyway, kind of, you know, one bright lining, I guess, of the kind of recent emergence of a major problem of CRB is it gives us a little bit of opportunity to use these to kind of do field experiments to evaluate lighting preference. So we kind of took these existing kind of programmable lighting and kind of designed enclosures for them, and then kind of put a diffuser on top to kind of make it more visible in a kind of omnidirectional pattern. So basically the diffuser is just a very thin shelled ping pong ball that we cut out and kind of attached to the housing. So you know, so we basically put these in the panel traps and kind of put them out, an array of 16 of them, so you know, different colors, and then kind of the lights mounted in different parts of the trap. So there's kind of two different options, one either up in the kind of top of the panel, kind of up, you know, in this left image, kind of where the pheromone lure is. And then we had an alternative mount kind of directly attached to the cup of these traps, and kind of, again, rotated these kind of different treatments, you know, with different lighting conditions and placements of the traps over a period of 16 weeks, and you know, basically evaluated the trap catches based on that. There is one error here. We've started kind of writing this up, and I actually kind of, when we retrieve these from the field and kind of looked at what was programmed, it was actually programmed to turn on for 6 hours after sunset and only 2 hours before sunrise, and no modulation. They just turn on and that's it, and then turn off at that given, you know, after that given interval after the sunset. So again, similarly, we kind of deployed all of these in a kind of a one small confined area in the Pearl City Peninsula, and thanks to Keith and his crew for putting those out and doing all the data collection and rotations. The summary, I'll spare you all the details, and you know, but if we pool all of the data and really don't try to look at the individual effects, the UV light is, you know, probably not a surprise, had a significant effect. So basically the large bars indicate attractiveness, or statistically a significant attractiveness of that particular color. So that's potentially kind of of use in kind of improving the effectiveness or efficacy of traps in the field. As Mohsen mentioned, he's been kind of sending them out to different islands to have deployed, again, to facilitate kind of early detection in the field. Related also to that, I think I need to wrap this up, but I think Mike Melzer, and this is a picture of Mike with our original aerial application platforms, kind of an agricultural drone. We kind of fit it to kind of apply spot treatments. Again, a very coarse, you know, orifice disc nozzle to just apply a steady stream of pesticide product directly in the crown. So he did some trials, and I'll kind of really race through these, primarily focusing on these pyrethroid products, mostly cypermethrin. The Demon Max and the Mustang Max are both cypermethrin products, and they're very effective. And we tested it out at some of these same locations that Cheng alluded to, so Ted Makalena Golf Course, as well as Hawaii Country Club. So some of the results, I mean, again, you know, very effective at pretty much instantaneous, not instantaneously, but, you know, over a very short, you know, short period of time, they basically drop right out of the trees. So again, when we apply these treatments and go kind of scout the field subsequently over the next couple hours, you know, find kind of dying beetles scattered all around under the trees, and, you know, again, quite a few. And so it's, you know, in addition to kind of a approach for kind of controlling the adult beetles directly in the crown, it's found it's also an effective way of actually scouting for kind of delineating where the populations are. You know, we kind of have a bunch of data, and again, I'll spare you all the details, kind of did some overhead aerial imagery before and after 3 months and so on. You know, evaluated some of the trap catches in the vicinity and kind of other control areas nearby. And essentially, you know, similar to what Cheng and Dr. Melzer kind of indicated, that, you know, it demonstrates efficacy and, you know, kind of anecdotal evidence based on the trees we've treated. I mean, it's kind of 3 months plus effectiveness at kind of still killing the adult beetles. I mean, people in those treated areas kind of report still finding dead beetles at the treated trees and not at other trees. We're kind of working on, again, as Dr. Melzer alluded to, kind of extending some of the SLN work and having this as a practice, regular practice. And also working on kind of authorizing kind of a cheaper drone to do this and basically modifying it. And so this is some video of that application. And that's basically it. Kind of quick acknowledgement. So definitely want to acknowledge Mohsen for being integral to a lot of this work, but I'll leave it at that. Thank you. Let's see, let me release the.
Thank you, Dan.
Yeah. Oh, shoot.
All right, so I see that the Q&A has been very active and our presenters have been answering a lot of those questions already. So if you have any more questions, feel free to type them in the Q&A box and we can go ahead and open up the floor if anyone wanted to talk about any of these answers. Okay, maybe everyone asked all their questions already. We'll give it a couple more minutes, see if there's any other questions that come in. Otherwise, we can wrap it up a few minutes early.
I reclaim my time. I'm just kidding.
Oh, here we go. Okay, yeah, if anyone wants to take these questions, any of the presenters, you can go ahead and just click the question that you want to take and share your answer.
Related to Jeff Burgett's question of exposure to tree trimmers, so I should, and I think it was in my presentation, again, our kind of protocol specified that fruit and flowers be removed prior to application. But, you know, I think the Mustang Max is labeled for agricultural use in tree nuts, and it has a post-harvest interval of 7 days only. So, I mean, again, I'm not an expert on the actual biology of it, but you know, so I mean, there shouldn't be an exposure for one, and at least, you know, if the EPA is doing their job right, it should be a minimal exposure for at least the cypermethrin products.
We have one question here from Elliot Parsons asking, what are the big gaps in advancing EDRR? Is there anyone that wants to take that question?
My apologies. Sorry, I was typing. What was the question?
Oh, what are the big gaps in advancing early detection and response? I think you're the best to answer that, Mike. Yeah.
Well, yeah, I guess you know, we know our traps are not the most effective. They're the ones that have been used, but and this is where everybody can get involved in identifying a better mousetrap. So probably having better surveillance. As Dr. Jenkins alluded to, their flight pattern, they don't fly a lot, so they don't have a lot of time to get into traps. But yeah, probably advancing trapping technology. A lot of work is trying to find additional pheromones that might be attractive at different times in their lives. So yeah, those are all approaches that need to be taken, and yeah, again, identifying a better trap is probably one of the better things that would help the program.
I see a lot of you typing. If it's easier to speak your answers, that's fine too. You can go ahead and just turn on your mic and answer them a little quicker.
I don't know, I feel it's usually best to just keep quiet, but to answer Anthony's question of how to get access to all the boring. Actually, never mind. Yeah. Yeah, with respect to drenching, so it can go's question. Yeah, we found, I mean, in our applications that definitely is very effective if you can really give a very precise drench right in the crown area where the boreholes are, and so and again, I mean, so pyrethroids are supposed to be very photosensitive, and so we're kind of surprised that they didn't photodegrade faster. But I mean, presumably, just kind of really getting into the tissues and notches in that area really helps extend, protect the product. And so we've had other collaborators apply on different islands and kind of different configuration and kind of a more traditional kind of flat spray arrangement, and it was pretty ineffective. And again, because it's not really targeted at where the beetles are, and it's kind of washing off of the fronds, and it's kind of in all cases not really appropriate.
All right. Since we're coming up on time, I see a lot of answers are being typed in the Q&A right now from a lot of our presenters. So while that's happening, I'm going to go ahead and launch one more. See, I'm going to launch actually a couple of polls. The first poll that we launched in the beginning ended too early. So if we can just do that one more time, that would be great. And then we're just going to do a quick 2-question post engagement poll right after that. So it's just going to take about a minute while we get the rest of these answers out. All right, we'll give that poll a few more seconds. All right, perfect. Thank you. Okay, and then we're still getting a few more questions. So we're gonna end the webinar in about a minute. So any last minute answers that we can get out there. I'm gonna launch one more quick poll. This one's gonna be really fast. Okay, I just ended that last poll. So we can go ahead and we're running a little bit over time. So we'll go ahead and answer a few more questions. And then if you have any other questions that weren't answered today, or you wanted to talk more or learn more about what was presented today, feel free to contact us and we can share your contact with the appropriate person. And we'll go ahead and type our information in the chat box for everyone.
So Keith, there was a question whether those 4,000 palm trees, which were trunk injected, were they labeled or tagged so that I guess it's for potential concern, right? Right. Mm-hmm.
Yes, the huge majority of them, the only times that we wouldn't tag is if it was on a private property and they said, I don't want to tag, but you can treat and I'll know that it's treated. But so if you have access to a tree, it should have a tag on it if it's in like a public area or something like that. But the huge majority of those got a silver tag that says CRB pesticide treated and then has a number on it. So they were labeled. There's a chance the tags can come off, but we generally treated all the trees in an area. So if you just kind of like look around at 5 or so trees, it's unlikely that tags have been removed from all the trees. there seemed to be a lot of questions about eradication, and eradication was attempted on Oahu, and a lot of the tools we're talking about today are new tools. And as Dr. Cheng told you, we had to do some testing to see if it was worthwhile doing some of these applications. So it wasn't until really about 2019, I think, when we started doing broader treatments. And by that point, there was a lot of infested area. So we were kind of behind the situation on eradication on Oahu. Also, the only successful eradication recorded on Earth was on a fairly small island, and they basically got rid of all of the breeding material, and it took them, I think, what, seven years to do that. So we're working in uncharted territory. There's not a known formula for an island like Oahu to achieve eradication. So we definitely have to develop some new tools to get a better control on things. Regarding other islands, we're doing everything we can to push back those populations, and nobody's promising eradication there, but of course we would love for that to happen. So yeah, we're throwing everything that we have access to, putting the tools that we think have the best chance of eradication forward on the other islands to see what can be done. So and so somebody mentioned start at the borders and move your way in. Yeah, I think we have to go zone by zone, because you can't just treat the whole island at once. And on Maui and Big Island, it's fairly geographically constrained. So I hope that answers people's questions.
Thank you, Keith. So we're actually coming up about 10 minutes over time. So we're going to go ahead and end this webinar today. There were almost all the questions answered, but if your questions didn't get answered, again, please feel free to contact us and we can route your question to the right person. I just wanted to say thank you again to all our presenters for coming, and for everyone that came and attended this webinar. If you would like to see the webinar or share the webinar, it'll be on YouTube. And then also we have another HiSam webinar tomorrow that you are welcome to attend, and we have the links for that in the chat. Okay, thank you all very much. I'm going to end this webinar now.